But now I will introduce our second speaker. That's Josh Tycko. And so Josh did his undergrad at UPenn, and then he spent 2 years as a research associate at the biotech company Editas Medicine. And then in 2016 started grad school at Stanford, where he is co-supervised by Bintu and Bassik. So Josh really has a stellar CV with many different honors prizes, awards, starting already during his undergrad when he was on the winning team in the iJump competition in 2013. He made it onto the dean's list, etc. But also during his graduate school, he was awarded both an NSF graduate research fellowship and recently an NIH F99K0. So that's all super fantastic. Josh's work has focused on high-throughput assays using CRISPR-Cas9, and so it seems particularly appropriate that he's presenting his work today while we honor two brilliant scientists for their role in discovering this CRISPR genome editing method. So I'm very looking forward to hearing about your work, Josh, and please go ahead.
Thank you for that introduction. And yes, I'm very excited about this prize that I just found was awarded to CRISPR today, which is really cool. So hi, my name is Josh. As she mentioned, I'm a student in the Bintu and Bassik labs at Stanford, and I'm excited to talk to you today about our project to discover and characterize human transcriptional effector domains using high-throughput screens. So we know that there are thousands of proteins that can localize into the human nucleus, and we'd like to understand which of these can function to regulate gene expression. For a protein to do this, we expect that it has to have at least two functions. It needs to be able to localize to its target genes, and this is commonly done through DNA binding domains in the case of transcription factors. And localization has been systematically studied with methods such as ChIP-seq. But the second function is carried out by effector domains, and these either directly or indirectly actually upregulate or downregulate the expression or transcription of nearby target genes. And effector domains have not been studied so systematically in human cells. So that's what we set out to do with this project. And the first realization we had that this might be possible is most of the domains in nuclear proteins are short. Specifically, we looked in the PFAM domain database, and we find that about 70% of the domains fit within our oligonucleotide library synthesis constraints, which in this case was 80 amino acids. And so what we were able to do is take all of the domains below this limit and print them on oligos and build a library. So we clone this library as fusions with a DNA binding domain called RTEDR, and then deliver it with a low dose of lentivirus to reporter cells such that most of these cells express one of these domain fusion proteins. We can then add doxycycline and induce the recruitment of a domain fusion protein to the genome, to this reporter that's integrated into a chromosome. And this reporter has a strong promoter, and then it has two parts, citrine for fluorescence and a synthetic surface marker that we engineered. So here's how the experiment works. In the beginning, day 0, the cells start out on. We then add doxycycline in the cells that are expressing a repressor domain that will be recruited, and over the course of 5 days, silence gene expression. And then to study the dynamics of this gene silencing, we then wash out the doxycycline and take additional time points. So we can see that some cells will reactivate while other cells will stay silenced with a permanent epigenetic memory. At each of these time points, we will mix the cells with magnetic beads that can bind onto the synthetic surface marker. And this gives us a practical and affordable method to separate the on cells that bind to the beads from the off cells that do not bind to the beads. We then just sequence the domains in the off and the on populations to compute enrichments and infer the effector function from those scores. And we call this method HT-recruit. So here are some of our results from a repression screen for all of the nuclear PFAM domains less than 80 amino acids. And I want to point out that this was a big collaboration between the Bintu and Bassik labs with many contributors to get it to the point where we have it working nicely. We can see good reproducibility across biological replicates, and we see hundreds of repressor domains. And if we look in the top right corner at the strongest repression domains, it included some, for example, some previously characterized crab domains that were known to work as repressors, which was encouraging. And we also see some repressors that had not previously been characterized, including some domains of unknown function, which are abbreviated as a DUF or DUF. So we then also take these additional time points. After removing doxycycline, we could see most domains do not give a permanent memory, and we'll see this reactivation and they start to move back towards the baseline, whereas in some cases we will see a longer term epigenetic memory depending on the domain. And I want to point out that this data is also filtered for just the domains that are well expressed in our cells, which we measure with a second high throughput method we developed that's based on the use of a flag tag. So to zoom in now on some of the hits, crab domains, these are the largest family of repressors. We had over 300 of them in the library and found that 92% function as repressors. Some of these had previously been characterized with individual recruitment assays. And actually there was a great paper in Nature Methods this week expanding that list up to 57 crab domains. And based on this previous work, we know that some crab domains can function as repressors and do this by binding a co-repressor called CAP1, or crab-associated protein 1, which in turn recruits histone modifiers and chromatin regulators that ultimately silence transcription. And also the CRISPR-I system is DCAS9 fused to the particular crab domain that comes from zinc finger 10. So in our screen, we found that some of the crab domains work as repressors and some do not. To better understand this, we integrated our data with some available co-immunoprecipitation mass spec results that came from full-length crab zinc finger proteins. We could see that the crab domains that do not repress in our screen are found in proteins that do not interact with CAP1. And the crab domains that do silence expression in our screen do come from proteins that interact with CAP1, with this one exception that was lowly expressed in our cells. And then we also looked at CHIP exo sequencing data, and we determined that the crab domains that are repressors in our screen come from proteins that co-localize on the genome in the same genomic locations where CAP1 can be found, while the others do not. Interestingly, we also saw a strong relationship between the evolutionary age of the crab gene and whether that crab domain could function as a repressor, wherein the more recently evolved crab domains consistently worked as repressors, whereas the more ancient crab domains did not, which opened up a question about what is it that these ancient crab domains are doing, which I will touch on later. But first I want to mention that we wanted to do an experiment to get a higher resolution look at how the sequence of the crab domain relates to its function as a gene silencer. And we did this using a deep mutational scan of a crab domain using the exact sequence that's found in the CRISPR-I system. So at every position along the crab effector, we made every possible substitution and then tested these mutants using HT-recruit. We could see these dark red stripes. These are residues where most mutations will abolish gene silencing activity relative to the wild type. And I think the pattern is even more clear when we look at triple consecutive substitutions. We can see it's really this middle portion of the crab domain that's called the A box, which is very necessary for silencing at day 5 of doxycycline recruitment. So if we then map where these residues are found onto a structure of that A box portion of the crab domain, we could see there was this periodicity where the necessary residues are found on a similar face of the helical structure of the A box. And these actually line up well with residues that are necessary for binding to Cap1 as determined in vitro studies. But we can get a fuller picture by taking additional time points in our screen. And so we could see that the A box was necessary for silencing, but this second portion called the B box was not so necessary at day 5 of doxycycline, but we do see some effects on silencing when we take a later time point at day 9. And so we're able to determine that the B box is not necessary, but does also contribute to the repression done by the crab domain. And then interestingly, we see in this N-terminal region, these dark blue patches. And so these are mutations that actually enhance silencing relative to the wild type crab. And this N-terminal region is interesting because it's very poorly conserved. It actually is spliced onto the crab domain from a distinct exon and is unique. This N-terminal extension is uniquely found in the crab domain from zinc finger 10. And so we were able to validate that mutations in this poorly conserved region gave us some enhancement in relative to the wild type. So the second largest family of repressor domains in our screen is the homeodomains. So homeodomains famously occur in Hox genes, these master regulators of development. And the Hox genes are really interesting in that they're organized on the genome in a simple way that corresponds with their behavior during development. So the Hox genes are found in these 4 clusters of genes, and the genes near the 3' end of these clusters are expressed earlier in development and also towards the anterior side of the developing embryo, whereas the more 5' genes are expressed later in development and at the posterior end of the organism. And what we were really surprised to see is that there's also a significant relationship between the repression strength of these homeodomains and their position in these clusters, wherein the more 5' genes, which are the later expressed and more posterior expressed genes, these are the ones that function as stronger repressors. So we're following up on this finding now, but we are speculating that this might be related to a really interesting phenomenon called the posterior prevalence rule, which states that when there are multiple Hox genes expressed in a given cell, the more 5' Hox gene tends to have a dominant effect over the more 3' Hox gene in determining the phenotype of that cell. And so we're now curious if this posterior prevalence might be related to the fact that these more 5' homeodomains are stronger repressors. So to get a better understanding of what might be causing these differences in repression strength, we took a closer look at the sequence of these homeodomains. And here we're looking at an alignment where we rank them in the order of their repression strength, with the strongest repressors at the top. And we could see that all of the strongest repressors contain this RKKR motif in the N-terminal region of the domain. So that's associated with the repression strength. We could also see that just the amount of positive charge in that N-terminal region is also significantly correlated with the repression strength. And here I'm just showing on an available structure of one of these homeodomains where this RKKR motif and where this positively charged N-terminal region appears relative to the more structured helical regions of the homeodomain. So far I've talked about repressors, but we can also use HT-Recruit to study activators. We do this just by changing the reporter to use a weak promoter such that the cells start out off. We can then add doxycycline to recruit an activator domain and turn the cells on. So when we tested the library with this reporter, we see that we can find activators which have a strongly negative log2 off to on ratio found in the lower left corner, and that there's good reproducibility amongst the hits, these activator domains, and that the hits contain some previously annotated activators such as the domains from the FOXO transcription factors, but also the hits contain several domain families that were not previously known to function as activators, and again, including some domains of unknown function. But one thing we were really surprised to see is that the strongest activator was actually a crab domain. It was one of these ancient crab domains that's very divergent in its sequence from the consensus of the modern crab domains and is more acidic, has more acidic residues, which is a characteristic that is commonly found in activator domains. So far I've talked about using HTRecruit with libraries that are designed around annotations of domains, which is based on sequence conservation or previous studies of domains. But we also wanted to be able to look for effectors in the unannotated regions of proteins. And so we did this with a tiling library where we take proteins and we chop them up into 80 amino acid tiles with a 10 amino acid sliding window. And we took this approach to study 238 proteins and discovered repressors in 141 of them. So this is an example from MGA. MGA is a member of the polycomb silencing complex, and it's a very large protein that previously had two annotated domains, which are both shown in gray and which were both related to DNA binding. But using this tiling approach, we can see these two peaks. So these are regions that contain repressors, which we just label repressor 1 and repressor 2. And so then we set out to validate these repressors. We could see they both do validate to silence the reporter, although they do so with distinct dynamics, wherein repressor 1 is a slower but more stable silencer, and repressor 2 is a faster but more transient silencer. We were then interested to derive the minimal sequence necessary for this effector function. And we did this by looking at the shared sequence between overlapping tiles found within these peaks. And taking this approach, we identified 10 amino acids that are sufficient for strong repressor activity from MGA1 and 30 amino acids that are sufficient for strong repression activity from MGA2. So using these approaches, we found effectors in many different proteins that come from diverse silencing pathways and complexes. And we were interested to see that they also then silence our reporter, but with distinct dynamics. And so we're excited that this should be hopefully a useful toolbox of very compact human-derived effector domains, which can now be used for further study and to build transcription perturbation technologies that will behave with distinct and tunable dynamics. So in short, what I talked about today is HTRecruit. This is our high throughput method for measuring effector function for both repressors and activators. We can use it on large libraries of domains or on tiling libraries across full-length proteins to discover effector domains. And putting these approaches together, we identify an effector in 600 human proteins. Once we have an effector in hand, we can then use HTRecruit to perform deep mutational scans and map the residues that are necessary for that effector function. And in some cases, like for this crab domain, we can also identify enhancing mutations. So we're excited to share this resource of effector domains, which we hope will be useful for both studying the biology of these transcription factors, building better models of how gene regulation functions, and also as components that can be used in synthetic biology as tools to perturb and manipulate transcription. So with that, I want to thank all of you for listening and thank my co-advisors, Lakra Bintu and Mike Bassik, who collaborated and really put the strengths of their labs together to make this project possible, and also major contributions from Nicole, from Galen, Aradhana, and from Anshul and his lab. And I'd like to thank funding, and I'd be happy to take any questions or feedback now, or you can follow up with me at these addresses. Thank you so much.
Fantastic. Thank you, Josh. Yeah, so reminder, you can type in your question in the Q&A or raise your hand. So I was wondering, Josh, for these new domains that you found, so do you know like how they would repress, or like are you planning to do like a CRISPR screen or something like a knockout screen in combination with it to like find out like what the mechanism of repression could be?
Yeah, so we definitely are planning that. That's a very good idea that I am hopeful that's going to work. We're also interested in using more biochemical methods to follow up on some of them, on some of the, especially the domains of unknown function, which I think may be some of the ones that you were referring to.
Yeah.
But yeah, I think that's a great idea.
Then there's a question from Jose Martinez. Great talk. Can you comment on how the subset of Hox homeodomains might be acting as repressors, excluding activators or recruiting repressors, etc.?
Yes. So this is just speculative, and we're doing experiments now to see if this hypothesis pans out. But we noticed that association with this RKKR motif, and also with an increasing amount of positive charge around that motif, and that made us think about nucleolar localization as a potential mechanism, because that motif and positive charge around that motif have been associated with nucleolar localization in the past. And so one idea is that these transcription factors might bias the region that they bind to towards being in a nuclear compartment that is relatively depleted for messenger RNA production. So we're going to look into that. And yeah, I will say that it's at least a little more complicated than that, because we find that that motif and positive charge alone is not sufficient to create repression when we find that motif in the context of other domains in our library. So it's probably, our thought right now is it's something about that motif as well as its context within a homeodomain.
Okay. So you mentioned one particular, like I think zinc finger 10, like crab domain that is now often used in CRISPR screens. Based on your results, would you make different recommendations, things that people should be using?
Yeah, I think so. For this preprint, we didn't do experiments where we fuse it onto dCas9, which is what you would really like to see. But we did find some mutations in this poorly conserved N-terminal region that enhance the function, and we saw by Western blot that they really enhance the stability of the fusion protein. So that might be useful in the CRISPR system. I think something I would also be excited about is that we now have many other very potent and short repressors that you could just use in combinations with the crab to get a stronger effect. So I think that might be a direction to go, but we haven't had much data on that yet. I would also say that there's this cool paper this week in Nature Methods that does test crab domains on dCas9 and finds that there's one of the crab domains they really focus on. They find that that works better than the zinc finger 10 crab domain for CRISPR-I applications in their hands. So that might be a good paper to look at if you want a better CRISPR-I system.
And I think Ben also had a question, and I think he can just go ahead and unmute himself.
Yes, great talk, Josh. I was asking about, so for the repressors, you had the RKKR motif. I was wondering, do you have any sense, or do you know if any of those get acetylated in the cell, and if acetylation of that motif would block the repressive activity?
Yeah, I think it's very possible, but we're following up on that now and do not have much to report there. But yeah, I'm excited to look into first just mutating regions of that motif to see which parts of it are necessary, and that might give us a hint to then look at how post-translational modifications at certain regions in that motif contribute to this function. So hopefully more to report soon.
Awesome, thanks.
Great. I think we'll wrap up there. Thank you so much, Josh.